Where's my pen? The nucleuses that are left behind are the supraoptic nucleus, and then we have this paraventricular nucleus, we have preoptic and anterior nucleus, and below we have this suprachiasmatic nucleus. Okay, so let's first see what about we have in this supraoptic nucleus. Okay, uh, now these are the things that you will be studying in this thing you will be studying in endocrinology. Okay, endocrinology. So but like I'm kind of telling you now because we are looking at this picture so that you may get a view of it. Now supraoptic nucleus synthesizes vasopressin, that is ADH, antidiuretic hormone. Okay, and paraventricular nucleus is responsible for oxytocin. Okay, so this is how you can kind of remember it, that I remembered it like this when I was studying SOAP soup. Okay, and then I was like that we have to, you know, skip one part. For example, if you're talking about antidiuretic hormone, then you have to make it this way and you have to make it this way. So like this I remembered that supraoptic nucleus with ADH and oxytocin with a paraventricular nucleus. So you can also remember this way. It is an easy way to remember if you're like mistaking it, but don't mix it. Okay, don't mix it like SO and AP. It is not like that. It is like SA and OP. Okay, you have to skip one in between. So this is how you can remember this, and you will study this more in the endocrine physiology. Then we have this control of parasympathetic system and the control of sympathetic system. So remember, if you remember this diagram that I told you, in this diagram I told you we have this hypothalamus, and this hypothalamus, when I draw it bigger, we have this two. One is responsible, the posterolateral one is responsible for the sympathetic, and the antero, or we can say ventro medial, is responsible for the parasympathetic. So you see they are also, these are the hypothalamus. Okay, so we have this control of parasympathetic system in the anterior nuclei, anterior one, and the sympathetic in the posterolateral. Okay, also they have said one thing which you have to remember is preoptic. This is different. Parasympathetic is also controlled by the preoptic. Okay, you know, the whenever your I also told you this thing yesterday, that we have this radial muscles of eye. Okay, we have these radial muscles of iris. Okay, and we have ciliary muscles in the side. Okay, so when these radial muscles constrict and ciliary muscles relax, our pupil dilates. Pupil dilates. That's what we called mydriasis. Okay, we call this thing mydriasis. So this mydriasis is a parasympathetic response. Mydriasis is a parasympathetic response, and sympathetic response is meiosis. Okay, the constriction of pupil is what we call meiosis. But now you see the receptors are present at this particular level of iris. We have alpha 1 receptors here. So these receptors are present at this particular level that are causing the pupil to dilate. But how the brain gets that signal is via this preoptic nuclei. So this is how we correlate things when we study neuro. Okay, so I think I'm clear to you. So similarly, I told you regarding the posterolateral. So the posterolateral was sympathetic. So just remember this diagram of hypothalamus. Okay, this is sympathetic and this is parasympathetic. So this is the posterior part, so posterolateral, and this is the anterior part. So you can say it anteromedial, or you can say it ventromedial. Okay, it has, you can say it is called in two names, anteromedial and ventromedial. So now you're done with you are done with all these nuclei. We have one nuclei left here. So let me tell you about it. Suprachiasmatic nucleus. This is very important. Very, very, very important. Okay. It controls the circadian rhythms. You will again study it in the cardiac physiology, but this is very important. Suprachiasmatic nucleus. Okay. Do not mix and confuse it with the optic chiasma. Okay. Usually people do confuse suprachiasmatic nucleus with the optic chiasma. Okay. That is just present behind this pituitary, and in certain tumors of pituitary tumors it is being compressed, and it gives you double vision. Okay, nystagmus, double vision, and all of that, the tunnel vision. So it is all because of the compression of optic chiasma. So people take it like suprachiasmatic nucleus must be dealing with vision. This is a misconception. I can say it misconception, but it is because lack of knowledge. Okay, this is purely lack of knowledge, because vision is not dealt with this. This is wrong. So you have to remember suprachiasmatic nucleus, it controls the circadian rhythms. That is a 24-hour biological clock that is going, that now at particular level you're sleeping, and you have noticed that when you wake up in the morning early, it is like you wake up at if you're going regularly on work routine, you're going on a job, you have to reach your office or your hospital at 8 o'clock, and you have to get up from bed at 6:30 or 6. So your mind already knows, and you wake up without an alarm. Okay, for example, you have these working days and then you got a holiday, but because you have been in a habitual routine of waking up at that particular time, you will wake up. Okay, and similarly for going to bed, you know that this is my time, and this is like I have to go to bed at this particular point. So you go to bed at that point. So you see these this is what we call circadian rhythm. This is the biological clock of our body. Circadian rhythm is a biological clock of our body. Okay, so some people, you are late a bit, Maria. We just finished this. Okay, I actually don't care about most of the people who come late, but there are a few people who are taking my classes from the first day, all of them, like 4 lectures in a day. And so we just finished the supraoptic paraventricular, preoptic anterior nuclei, and posterior lateral nuclei. Okay, so you kind of missed this part because you're late. Okay, the next you will get the recording at night probably, or I'll try to upload it in the evening if we got free early today. So now you're clear. You see how little it is written, but how much you have to go through and understand this thing. Now you will always remember the hypothalamus nuclei, the midbrain, pons, and medulla oblongata. So this was one topic of our lecture today, which is completed. Now I will move to the second topic. We have set 3 topics. So our second topic, okay, physiology lectures will be a bit lengthy, because I am not teaching pathology together with physiology. Maybe if on Saturday and Sunday, we do not have class on Saturday and Sunday, but if I'm like if I'll check my schedule. If we'll have time, I will put in one class for pathology, okay, for general pathology. So yes, maybe on Saturday or maybe on Sunday, we will have one class for general pathology, so that we may cover general pathology before starting systemic pathology. Once we will finish this neurophysiology, the next topic will not be of physiology. The next topic will be of pathology and neuropathology, okay? So I will take physiology and pathology together, like neurophysiology, then neuropathology, cardiophysiology, and then cardiopathology, because when you correlate physiology and pathology together, it is easier for you to understand, okay? And both the books of physio and patho are BRS, so it will be more easier for you to grab the concept from those books. But I want to finish at least 2 chapters of general pathology before starting neuropathology, because they will make your base. Okay, now the next topic which is given here in sensory system in general, we have to, I will like kind of teach you this portion today. But before moving on to the sensory systems, in the 6th edition we do not have there was another topic given after this in my 5th edition book, okay, and that was organization of central nervous system. So I will teach you it from my notes, okay? I will teach you it from my notes. Acha, before moving further, I want to show you one more thing. These notes are like completely totally covered now, okay? These were like more about basic anatomy. Okay, so now this was the topic which we were studying from the book, okay, and I just wrote a bit of it for people who do not understand the concept of hypothalamus, okay? If somebody do not know the concept of hypothalamus, that what is a hypothalamus is and where it is situated or where it is located, I have this picture, I think, wait a second, yes, it is here. You see this? You see this is the pituitary gland, this is the infundibulum, okay, and this portion is the hypothalamus. This portion is the hypothalamus, this is the pituitary gland, and this is the thin stalk which we call infundibulum, okay, and this is the ventricle, this is the third ventricle, fourth ventricle. So this whole portion is what we call limbic system, okay, and this is hypothalamus. So hypothalamus is basically a part of limbic system. I am repeating this again for people who are new, okay, but I have taught you this all in the first lecture. So I want to take you to this area. I'm sorry for scrolling. Acha. You see, this hypothalamus is a part of limbic system which consists of subcortical and cortical components. When we talk about subcortical, we see hypothalamus because cortical is like cerebral cortex. If this is the brain, the upper portion is the cortex, and below this is what we have subcortical region, and that subcortical region has this limbic system. Okay, so this hypothalamus is a part of subcortex, okay, and then what it contains, what it has is septum para-olfactory area, anterior thalamic nuclei, posterior thalamic nuclei, okay? There are certain nucleuses which we just saw, like supraoptic, paraventricular, the nucleus is for thirst and temperature, the nucleus is for parasympathetic and sympathetic control, okay, all these things. Then we have a hippocampus over here, which is very important, okay. We have a amygdala, it is also very important. We have basal ganglia, it is also very important, okay. We have portions of basal ganglia. So if you are like kind of, do not worry about these three little things names. If you're new, go back to my first lecture, and if you're following from the first lecture to the third lecture, you will not have trouble understanding these three verbs. Okay, so if I'll repeat it again and again, then it will be really hard to cover our course in 6 months. So I'm just kind of telling you. So this is how we'll roll on. Now the next topic for today is this nervous histology. Okay, now the next topic for today is nervous histology. Basically, I can say it more than histology, it is physiology. Okay, here I will tell you first of all about, because before moving further, receptors and everything, you should know that what is the basic component of your nervous system, so that before moving to sensory and motor systems, you may understand what these cells of these nervous systems do. Okay, and the main functions, like I'll not go in a lot of detail, uh, but I will kind of these notes are in a bit detail for the people who have no idea and no concept, like zero concept. Okay, so these are for them. And but if you have it, you can like just give it a read and take out the important points from it, and you can like uh, put it in. Okay, so our nervous system is composed of two types of cells, these nerve cells which we call neuron, and these glial cells which we call neuroglia or glia. Okay, but these glial cells are also, you know, subdivided into four. We have four types of glial cells. Now what are they? Schwann cells, oligodendrocytes, ependymal cells, and astrocytes. So, you know, Schwann cells, oligodendrocytes, ependymal cells, and astrocytes. So these are very important to know. When we talk about this, what we say when we talk about this nervous histology. Okay. Acha. So, so when we like kind of move further, you see this the function of neurons is they gives the sensory stimulus, in motor that is response. In case of response, so stimulus is sent by the sensory part, and the motor neurons are responsible for the response. Okay, and then the reflex action you perform. They conduct impulses, they store information, they retrieve information. So if you remember from our first class, I told you about the integrative function of the brain, that whatever information is processed in the brain is you know whatever information is processed in the brain, only 1% of the information is used by the brain and is taken as the memory. The rest of it is just thrown out. The brain says that we don't want this information because this is not important. So neurons also store information. Okay, all that information, that 1% information that the brain takes seriously, is stored in the form of memory. Okay, so one function of neuron is storing information and another is retrieving information. Then definitely conducting impulses via sensory neurons, that is carrying the stimulus, and the response the central and peripheral nervous system gives us to our effector organs is via this motor neurons. So these are the little function of neurons. Okay, then we move further to the structure. Now I will teach you this structure from this picture. Okay, I will not read line by line from these notes, and you can have these notes. I will put them in the drive, but for now, like you, I will make you understand this from this picture. Now you see, this is This is what we call soma, or we call it cell body. Okay, and these are dendrites. So these dendrites are not straight, these are like this. Okay, and they can be multiple in number. Okay, one this kind of dendrite, one dendrite can receive 9 synapses. Okay, one dendrite can receive 9 synapses. Why they're like this kind of present in this way? To increase the surface area. Okay, that's what we have studied in gut also now. Okay, small intestine is, present like this, so that it can increase the surface area for absorption. So same goes with the dendrite. So one dendrite can receive 9 synapses at one same time in the central nervous system. So this is an important point to remember. In this soma, we have this nucleus. Okay, and definitely surrounding this nucleus, definitely this is a cytoplasm of a neuron, right? So the cytoplasm of neuron, now there are some little terminologies that are very important to remember, in case of they don't use the word cytoplasm of neuron, so what will they use? So the cytoplasm of neuron, in contrast to this, they can use the word nasal substance. It is written over here, nasal substance. Okay, they can use nasal substance. Nasal substance is the cytoplasm of neuron. Okay, and there is this region which we call axon hillock. It is this triangular part which do not contain nasal substance, that is, it does not have cytoplasm. Okay, so just keep this thing in your mind, that nasal substance is the word used in replacement to the cytoplasm of neuron. Okay, then we have this whole neuron is covered by a membrane. This whole neuron is covered by a sheath of membrane. So that sheath of membrane is called neurolema. It is not written over here, but you can write it in your notes. It is called neurolema. This is basic physiology and structure of neuron. You should know it. Neurolema. What is neurolema? It is the membrane of neuron. Okay, membrane of neuron. Cytoplasm of neuron is nasal substance. Membrane of neuron is neurolema. Then we have this nerve fiber, which we called axon. So axon is also used in replacement of nerve fiber. Okay, they can use the word nerve fiber in MCQs in replacement to axon. So you should know what they're talking about. Nerve fiber, they're talking about the axon of neuron. Okay, and then the soma can be used in replacement to cell body. Soma is basically a Latin word for cell. Okay, so cell body. So please remember these little terminologies where students make mistakes. Okay, now clearing all these. Now at this particular region, you must have endoplasmic reticulum. Okay, you must have mitochondria, because this is a cell. Okay, it must have all these components. So in the cell body, in the soma, we do see the production or the maturation of proteins. Okay, so it takes place in the cell body or in the soma of neuron. Okay, Acha. Moving further, then I told you about the dendrites. Okay, I told you about this axon hillock, the triangular portion that lacks the cytoplasm, or we can say that lacks the nasal substance. Okay, then moving down, we have this axon. Okay, or nerve fiber, and on this axon there is a sheath. There is a sheath of fat. This is fat. Myelin sheath is basically made up of fat. It is white in color. Okay, this myelin sheath is present over here to protect the flow of impulses out. Okay, ताकि जो impulse आ रहा है वो सीधा जाए. The impulse should go straight. Okay, it does not jump out. And then they leave little portions in between these myelin sheath, from where the impulses can jump from one to another. So you have to remember this myelin sheath. You can take it as an example for a wire. You seen the wiring? There is a copper wire, and then there is a plastic that is covered above that copper wire. So that plastic is basically myelin sheath, and that copper wire is this axon which is running uh, beneath the myelin sheath. Okay, so you can take this as like that. So there is a condition in which this myelin sheath is destroyed. We call that demyelination of myelin sheath. Okay, and that disease is called multiple sclerosis. We will study this disease in neuropathology. Okay, just for now, you have to remember this thing. See, myelin is a lipoprotein sheath on axon. The more myelin we have, the more rapid impulse it will be. So more myelin, more faster impulse. Okay, it is a lipoprotein sheath that is made up of fat. And then I told you about the soma, the cell body. So you see the cytoplasm, the nasal substance, have this endoplasmic reticulum, the rough endoplasmic reticulum with protein synthesis. Now, if you have like a good concept of cell, the endoplasmic reticulum, we have smooth and rough. The rough endoplasmic reticulum have ribosomes on it, okay? And those ribosomes are basically responsible for protein synthesis, okay? So let's see what else they offer. Now this is something which I've told you, that they conduct impulse away from the soma, dendrites conduct impulse to the soma, and then we have short and long axons. Okay, so the location of short axons are found in the granular cells of cerebellum and the granular cells in dentate of hippocampus. We have this third ventricle, and then at the sides of it we have a dentate nucleus. Okay, so this region is called hippocampus. So olfactory epithelium new cells normally regenerate. Okay, the word is not written over here. They regenerate every 40 days. This is just a general knowledge. Usually we say nerve cells don't divide after birth, but they are places where they can divide. So the short axons actually do divide. Okay, and they are present in the olfactory epithelium. So the sense of smell, the olfactory epithelium in your nose, the short axons are present there, and they regenerates after every 40 days. So this is some journal knowledge for you. Then you see this long axons, they are present in the cerebral cortex. Okay, and from the cerebral cortex, they move to different parts of the body. Okay, and also in the anterior horn cells in the spinal cord. Now you see in the spine, in this for example, this is a spinal segment, and we have this butterfly shaped structure present over here. Okay, so this butterfly shaped structure, this is the gray matter. Okay, and the rest of the area is the white matter. So it has horns. Okay, this is the posterior or the dorsal horn, this is the anterior horn, and this is the lateral horn. So the long axons are present over here in the anterior horn, at this in these horns. These are the anterior horns. Okay, so this is some general knowledge for you again, that the polio virus attacks anterior horns, ultimately on risk to paralysis. So you know, it will it's just a general knowledge, it can help you in microbiology. Then what else we have? Neuroaxon has a neurotubule and it has a neurofilament. Okay, it provides a structure, help direct exoplasmic flow from soma to synapse. That's what I have already told you. Um it carries impulses. Now here comes, also I told you regarding this dendrites. Okay, dendritic spines increase surface of dendrites and it can receive as many as 9 synapses. Okay, they can change their structures in every 30 seconds. So this is all about something about dendrites we have. And now these things are important. So before the lecture ends, I just want to complete this part today, that we have, now this was all about neurons. We have neurons and we have uh, glial cells. Okay, so the glial cells are basically 4 in number. One of them is Schwann cells, one of them is oligodendrocytes. Okay, another is astrocytes, and number 4 is ependymal cell. Okay, so we have 4 of them. So the Schwann cells are basically the ones who are responsible for myelination of the peripheral nervous system. Remember this MCQ is favorite. It is a favorite MCQ. Schwann cells myelinate the peripheral nervous system, that is the nerves arising from the brain, that is cranial nerves, cervical nerves, thoracic, lumbar, sacral, all these nerves, peripheral nervous system. Okay, and the nerves present inside the brain, inside the spinal cord, they are myelinated by oligodendrocytes. So these are important MCQs. Mug it up. Okay, याद कर लो अच्छे से. How I remember it by the name Shan. You know, Pakistani, we have this Pakistani hero, actor Shan. So P for Pakistan, and I remember it that way. You can also remember it the same way. So if you remember one, you will remember the second one automatically. Then you see this picture. This is a very good picture. These are this is the neuron of peripheral nervous system. Okay, this is the neuron of peripheral nervous system, and on this axon we have this Schwann cells that are coiled. So this is one Schwann cell and it has this nucleus. This is another Schwann cell. This is another Schwann cell, and likewise, this is not 1, 2, 3. This whole is one. This whole is one, and they have this nucleus over here. And these Schwann cells, basically that lipoprotein layer which we called myelin sheath. Okay, and beneath these Schwann cells, one Schwann cell and two Schwann cells, we have this little area which is left, which is non-myelinated. Okay, this area is called node of Ranvier. Okay, it has ion channels over here. Sodium ion channels are present over here. Okay, so the conduction that jumps from here to here to here, this type of conduction is called saltatory conduction. So you can like write it in your notes. This conduction that jumps from one node of Ranvier to the other node of Ranvier, this conduction is called saltatory conduction. Okay, so um, just remember this. Then some things are not written over here, so like I'm just telling you so you can write it, because these words, when they come in the MCQs, students don't understand. Okay, now this point is really important. You see, we have lots of Schwann cells that are myelinating one axon. But when we talk about the axon of the neuron of central nervous system, the neuron of central nervous system, its axon has only one oligodendrocyte. So only one oligodendrocyte will myelinate the axon of central nervous system, but too many Schwann cells can myelinate the axon of peripheral nervous system. Okay, so this is the difference. Then we have astrocytes. Okay, So about astrocytes, you have to remember that they are, okay, these two are macroglia and this is microglia. So you have to, the thing you need to remember regarding the astrocytes is they form blood-brain barrier. So this is important for your PMDC MCQ. Astrocytes form blood-brain barrier, okay? There are other functions also written over here. They are star-shaped, they initiate the formation of blood-brain barrier, they have tight junctions and glial membrane. Okay, this picture, just see this picture, you will understand in this picture. You see these are astrocytes and this is pia matter. So their projections are reaching at the pia membrane, okay, the foot processes. So it is for the protection, and when there is excessive neuronal firing, the astrocytes pick up excess potassium. So this is another point which, you know, many people will not tell you. So you have to remember this point as well. There are two functions which are very important regarding astrocytes. One is they form blood-brain barrier. Another is after neuronal firing, they pick up the excess potassium. Okay, so this is how they're located. Then you should know regarding the okay, oligo is given over here. Then you should know regarding the ependymal cells. It's not mentioned over here. The ependymal cells make up the blood CSF barrier. Okay, I will send you the picture of book if it is not in the PDF. So you can just write it in your notes that ependymal cells makes the blood CSF barrier. Okay, they are present at the ventricles. We have this third ventricle, fourth ventricle, we have this choroid plexus present inside. So they just line at that particular level. So they line the blood-brain barrier and the CSF, blood CSF barrier, basically, blood CSF barrier, ependymal cells, okay? So at this particular point, if I, let me have this picture. I don't know if I have it right now, but I'll send you the picture in on WhatsApp, okay? I'll send you that picture on WhatsApp. I've not taken the image, so you can see. But for now, you can just write down in your notes that it makes up the ependymal cells makes up the blood CSF barrier, and astrocytes makes up the blood-brain barrier. Okay. Now, the only topic for in the cells which is left is microglia. Okay. Now, let's move to microglia. This is oligo, this is microglia. Okay, regarding the microglia, this word is important. It has this phagocytic effect. You know, macrophages, the macrophage of brain is microglia. So this is a simple MCQ that is asked in the NDC exam and other exams also, that the macrophage of brain, of nervous system is microglia. This is the macrophage. Okay, they proliferate when there is a neuronal injury. Okay, and then they serve as scavengers. Okay, they eat up all the cellular debris that is remaining. The brain is injured, there is now debris in the brain, so all that cellular debris is being eaten up by these scavengers, which are microglia. They are the macrophages of brain. Okay, very small until activated by injury, when they become phagocytic. Okay, for example, in a disease like Alzheimer's or other diseases, okay, many microglia cells are present. Okay, then you will see in the, when you will see this scans, you can, when you will do it at the cellular level, okay, microscopic, you will see this brain biopsy is also being taken, right? So when in the biopsies, you can see this lots of microglia in that particular case. So I guess I guess for today, we have studied these cells and also the main portions. Now, for tomorrow, we will kind of begin the sensory receptors. I actually wanted to touch. I will teach you the synapse and other things with the action potential that is remaining. Okay. So, but I want to teach this a little thing. I want to just start this sensory receptors for now. Okay. Um, but this is not given so good. Okay, so maybe we can do this tomorrow. I want to show you two charts. Okay, there are like very good charts which can differentiate these type of receptors, so it will be easier for you to learn it this way. The way it is given in BRS is not that good. Okay, so maybe I'll put in those charts tomorrow, and we can continue with the sensory receptors and further on tomorrow. This is a very important lecture which is remaining. Okay, we have to study these nerve fibers, their types. Now I am telling you that in the PMDC exam, these Pacinian corpuscles, Ruffini, and you see Ruffini is not written over here, Pacinian, Meissner's, Ruffini, all these are very important. They are asked repeatedly in exam. Also, did you ask about these nerve fibers, the conduction? Okay, this chart is very important. And then this is a long topic. Sensory system is a long topic. We will go like one by one, and first order, second order, third order neurons. A bit about action potential comes in here. Then somatosensory system. Okay, all the details of these nucleuses. So these things will take a bit time. Neurophysiology is time consuming. Learn this chart. This is very important. Then we have vision. It's a long way to go still. We have a whole lot sensory system. Then we have a whole lot motor system. So, but don't worry, we will be studying from the book completely. And then we have this motor system. So just just take the classes every day. That's all I can say. So let's finish our lecture on here, and then I will be having it as 5:10. I will be having some break by 5:25 probably, and then we will start the class for I guess neuroanatomy. Okay, not neuroanatomy, sorry. uh, we are doing upper limb anatomy. Okay, so today we have to cover muscles of upper limb anatomy. So I will be teaching you upper limb anatomy, or I will teach you pharma first. So let's see. I'll kind of figure it out in next 15 minutes. So I will see you people at 5:30 p.m. now. From it is 5:11, and we will start the class at 5:30 to 6:30. Okay, the next class. Thank you so much, and I'm just ending the lecture here.